My name is Madeline Totten. I'm a second-year BMS student, also working with Dr. Lowry, and my project is on the antagonism between the bone morphogenetic protein, aka BMP, and activin signaling pathways in osteoprogenitor cells. So a little background information. You heard a little bit about osteoporosis from Kelly, so we know that osteoporosis-related bone loss occurs when the rate of bone resorption exceeds that of bone formation during the bone remodeling process. And we know that the BMP pathway on the left promotes bone formation in osteoblasts, while in the activin pathway we see bone resorption. So there's studies that show that there's antagonism that occurs between these two pathways, but there's not a whole lot of research on the molecular mechanism behind all of it that's allowing for this interaction. So a more detailed understanding of this molecular relationship is needed to provide insight into potentially new therapeutic strategies for osteoporosis. So we believe that these pathways interact in a seesaw manner, so one pathway would increase at the expense of another. Kelly's project showed you that when the type 2 BMP receptor, BMPR2, is knocked out, we see the increase in bone mass due to BMPs being able to utilize that shared ACVR2A receptor. Studies show that these BMP and activin ligands compete with one another for those shared receptors, and these receptors are the exclusive type 2 receptor for the activin ligand. We actually see that there's a slightly higher affinity in normal cells, that activin has a slightly higher affinity for those receptors than BMP does. So we kind of just wanted to further study this competition that's occurring between the two, where and why it's occurring. And to start that, we decided to inhibit parts of these pathways in different locations and see what the downstream effects are. So first, we wanted to see what would happen if we sequester the activin ligands before they can engage with their receptors, so extracellularly sequester them. And we use an activin binding protein called follistatin, which binds to the activin ligands and neutralizes their effects. We predicted that if these activin ligands are sequestered, due to the seesaw interaction between these two pathways, we predicted that there would be an increase in BMP signaling if activin is not allowed to signal. This result is shown in the Western blot here. The activation of the pathways is measured by the phosphorylation of either SMAD158 or SMAD23. So with follistatin treatment, we see an increase in the phosphorylation of SMAD158, which tells us that more BMP signaling is occurring when the activin ligands are sequestered. So this was our first evidence that told us that yes, these pathways do interact in a seesaw manner. And it showed us that under endogenous circumstances, activin is somehow repressing the activation of the BMP pathway, which is why when we take activin out of the picture, we see an increase in that. So next we wanted to see, does this also happen on the other side of the seesaw? So we treated the cells with an extracellular antagonist called noggin, which is specific to BMP ligands. It sequesters them, again, upstream of receptor engagement. And again, we predicted, okay, if the ligands are sequestered, BMP signaling cannot occur, we should see an increase in the opposite side of the pathway. Interestingly, though, we did not see any change in the amount of activin signaling that's occurring. And we knew that this wasn't a technical error because noggin did completely block BMP signaling from occurring. We then wanted to see, So at this point, we kind of just gathered our thoughts and said, okay, we know activin is repressing BMP signaling in normal circumstances, but it doesn't occur on the opposite side. So to kind of look more at that repression, we wanted to see, is this occurring outside the cell or inside the cell? So we really wanted to find a way to separate these extracellular events from intracellular. So we did that. We were able to block activin signaling using a drug called SB431542, which binds to the activin type 1 receptor and blocks it from being active, so activin is no longer able to signal. And unlike the signaling, so when we used follistatin, we saw the increase in BMP signaling. With this drug, we didn't see any change in the BMP signaling. And we, again, knew this was not a technical error because this drug did stop activin signaling from occurring. So a summary up to this point, We know that follistatin sequesters the activin ligands extracellularly before they can interact with their receptors. And when that happens, we see the increase in the BMP signaling. With noggin, which sequesters the BMP ligands, we do see that it stops the BMP signaling from occurring, but it does not have any effect on the activin signaling occurring. And the SB431542 blocks the activin receptor at the cell surface level, stops the activin signaling from occurring, but again has no effect on the opposite side. So our working hypothesis at this point is that the activin-mediated repression of BMP signaling is ligand-dependent and occurs upstream of effector engagement via competition for a shared pool of receptors, specifically those ACVR2 A and B receptors that we see are used by both ligands. So we came up with a couple of specific aims and future directions at this point, one of which being, can we alleviate this competition between the ligands if there is an abundance of this shared receptor? And two, can we neutralize the activin ligands similarly to follistatin using specific neutralizing antibodies targeted to activin? So for the first specific aim, we wanted to somehow be able to overexpress the shared ACVR2 A and B receptors in our cell line. So we first tried a method called transfection, and one of the medical students in Dr. Lowry's lab spent a whole summer trying that method and had no luck. So then we tried a method called transduction using an adenovirus, again, had no luck. And then I had actually been working this summer using a lentivirus for transduction with a different project. So that's another plug for getting involved in multiple projects and honing your skills to get on other publications. So we decided to try to use a lentivirus for this cell line. And so we used a lentivirus that had a GFP tag, which is green fluorescent protein. So when you transduce, transducing, you treat the cells with this virus. The virus has a plasmid containing your gene of interest. So for this one, our gene of interest was GFP. So if the cells were successfully transduced, we should see it's green fluorescent protein, so it fluoresces under microscopy. And you can see in this picture that the green cells indicate that, yes, the virus did get into these cells. So next we wanted to keep the GFP cells going as our control, and then we used a lentivirus that had the gene of interest of the ACVR2 overexpression. And it's a lentivirus that had cDNA encoding C-terminal V5 tagged human ACVR2A. So we transduced the cells, again, with that viral strain. So both of these viruses are puromycin resistant, so we treated the cells with puromycin, and so that means any cells that weren't transduced would die, and the ones that were successfully transduced would remain alive. So then we ran Western blots to confirm the overexpression of the lentivirus. So since our ACVR2A virus had a V5 tag, we first did a Western blot with an anti-V5 antibody just to show that the cells had this gene inside of them, and that you can see here. And then we did a Western blot against the ACVR2A antibody to confirm overexpression, and you can see compared to the control cells, it's vastly overexpressed in these cells. So then our next step was to repeat that follistatin treatment with these cells overexpressing this receptor. So we predicted that if the shared receptor was the limiting factor in the competition between these two pathways, then flooding the system with tons and tons of this receptor should eliminate the competition. So an analogy to kind of explain this in easier terms is that there's a lot of BMS students trying to get into medical school, and in normal circumstances, there's a limited amount of spots, and that the spots are like receptor availability. And when we, so the ligands are the BMS students competing for this receptor. There's not a lot of spots, not a lot of receptor, and it's with varying affinity. So we think that if we can flood the system with more receptor, aka more medical school spots, that would eliminate the competition between the students, and we would not see that anymore. So when we ran our Western blot, we did see that, so the GFP is our control cell line. We did see that same increase in BMP signaling when we treated with follistatin as we saw before. But in the cells overexpressing this shared receptor, there's no longer a competition occurring because of the fact, like, we're not seeing that there's an increase in BMP signaling, which tells us that the competition is eliminated. And the Western blot gods are kind of hating me right now, so I haven't had the opportunity to check for activation of the activin side of this. So we do want to repeat this experiment again and check for phosphosmad 2-3 levels. But we wanted to see kind of where we're going with the project and what we've been able to find so far. So for the specific aim 2, the neutralizing antibodies, our goal is to find a neutralizing antibody that can act similarly to follistatin in normal cells. So if we could have something targeted to specific activin ligands that can lead to an increase in BMP signaling, that could be potential for drug development down the road. So activins can have different subunits. So we first did an RT-PCR and gel electrophoresis to confirm which activin ligand, or not activin, subunits, sorry, that our cells have. And we found out that our cells have both activin A and activin B. So we plan to treat these cells with neutralizing antibodies against both activin A and B. And then we predict that, similarly to follistatin, we would see that when these cells are treated with these antibodies, there would be an increase in BMP signaling. And if we get these results that we're predicting, we would hopefully be able to move to an in vivo study where we could treat mice with these anti-activin A or B antibodies and then look at the effects on bone mass. There's also in vivo studies that show if you treat mice with a receptor decoy for ACVR2B, it sequesters the activin ligands, and we do see a reduction in activin signaling and an increase in bone mass. So we're predicting, similarly to the results from that receptor decoy, if we treat mice with these neutralizing antibodies, we would hopefully get the same results. So to conclude, we know that BMPs and activins interact in a seesaw mechanism, with one pathway increasing at the expense of the other. BMPs and activins compete for the shared type 2 receptors ACVR2A and B, and activin-mediated repression of BMP signaling is ligand-dependent and occurs upstream of effector engagement due to competition for those shared receptors. I would like to thank my mentor, Dr. Jonathan Lowry, and then my committee members, Dr. Lillian Plotkin and Julia Hum, the funding that makes this research possible, and the Bone and Mineral Research Group. And I will take any questions.